Okay. So before that, I just wanted to discuss regarding, I think you all watched the status of Prasanna sir, or the YouTube link regarding one of the contest organized by Sankalp Learning, that is Sankalp Edu Hamp ಅಂತ ಗೊತ್ತಾಯ್ತಾ? ಸಂಕಲ್ಪ ಎಜು ಹ್ಯಾಂಪ್ ಕಾಂಟೆಸ್ಟ್ ಇದೆ, ಎಸ್ಪೆಷಲಿ ಫಾರ್ ಸಿಇಟಿ ಆಸ್ ವೆಲ್ ಆಸ್ ನೀಟ್ ಸ್ಟೂಡೆಂಟ್ಸ್. ಯಾ, ಇದರ ಬಗ್ಗೆ ಇನ್ಫಾರ್ಮೇಶನ್ ಇದೆನಾ ಐಶು ಅಂಡ್ ಕೋಮಲ? ಸಂಕಲ್ಪ ಎಜು ಹ್ಯಾಂಪ್, the YouTube link has been shared today morning. Yeah. So regarding that, if you are really interested, you all can participate in it. And we have much more, what we can say, plans or strategies for the board exam as well as for the NEET exams and CET exams too for those students. So try to crack it. Fine. So I hope to see you all in that fresh new batch as EduHamps. So as we were discussing regarding the principle of inheritance, linkage and recombination we discussed. Yeah, polygenic inheritance we discussed. Now we have the other point, which is called pleiotropy anta. So ಈ pleiotropy ಅಂದ್ರೆ ಏನು? A single gene can exhibit multiple phenotypic expression. Such gene is called as pleiotropic gene ಅಂತ. ಸೊ ಇಲ್ಲೇನಾಗತ್ತೆ, ಒಂದೇ ಜೀನು ಮಲ್ಟಿಪಲ್ ಫಿನೋಟೈಪಿಕ್ ಎಕ್ಸ್ಪ್ರೆಷನ್ ಅಂದ್ರೆ ವೆರೈಟಿ ಆಫ್ ಕ್ಯಾರೆಕ್ಟರ್ ಅನ್ನ, ಒಂದು ಎಕ್ಸ್ಪ್ರೆಷನ್ ಅಂದ್ರೆ ವೆರೈಟಿ ಆಫ್ ಎಕ್ಸ್ಪ್ರೆಷನ್ಸ್ ಅನ್ನ ಏನ್ ಮಾಡತ್ತೆ, ಎಕ್ಸಿಬಿಟ್ ಮಾಡತ್ತೆ. ಸೊ ದೋಸ್ ಜೀನ್ಸ್ ಆರ್ ಕಾಲ್ಡ್ ಆಸ್, ಸಚ್ ಕೈಂಡ್ ಆಫ್ ಜೀನ್ಸ್ ಆರ್ ಟರ್ಮ್ಡ್ ಆಸ್ ಪ್ಲಿಯೋಟ್ರೋಪಿ ಅಂತ. ಹಿಯರ್ ಅ ಸಿಂಗಲ್ ಜೀನ್ ಕ್ಯಾನ್ ಎಕ್ಸಿಬಿಟ್ ಮಲ್ಟಿಪಲ್ ಫಿನೋಟೋಪಿಕ್ ಎಕ್ಸ್ಪ್ರೆಷನ್. ಅದರಲ್ಲಿ ಮುಖ್ಯವಾದದ್ದು ಅಂದ್ರೆ ಇಲ್ಲಿ ಫೀನೈಲ್ ಕೀಟೋನ್ಯೂರಿಯಾ, ಫೀನೈಲ್ ಕೀಟೋನ್ಯೂರಿಯಾ. So that is the example for pleiotropy. What is this? This disease is caused due to mutation in the gene that code for the enzyme phenylalanine hydroxylase. So one the gene ಇದೆ. So that gene will code for a enzyme which is called phenylalanine hydroxylase. But due to mutation of genes, phenotypic expression will be characterized by mental retardation, reduction in hair, reduction in skin pigmentation. So ಈ ಒಂದು gene ಅಲ್ಲಿ mutation ಆಗಿ, what do you mean by mutation? A bit change in the gene. A bit change in a gene is termed as mutation. Regarding that we will learn later. So ಆ ಒಂದು ಚೇಂಜ್ ಆಗಿ ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ, ಫೀನೈಲ್ ಕೀಟೋನ್ಯೂರಿಯಾ ಅನ್ನೋ ಕಂಡೀಷನ್ ಕಾಣಿಸ್ಕೊಳ್ಳುತ್ತೆ. ಸೊ ವಾಟ್ ಈಸ್ ದಿಸ್ ಫೀನೈಲ್ ಕೀಟೋನ್ಯೂರಿಯಾ ಕಂಡೀಷನ್ ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ, the children will be mentally retarded, the hair will be very reduced hair irutte, then skin pigmentation will be reduced. So next we have sex determination anno concept ide. So this sex determination anno concept anna Henking, the scientist named Henking in 1891 traced a specific nuclear structure during spermatogenesis of some insects. Wow. So when he was studying regarding certain insects during spermatogenesis, that is during sperm formation in certain insects, he observed a specific nuclear structure. And 50% of the sperm received these specific structures, whereas 50% sperm did not receive it. So 50% of the whatever sperms formed, they have that specialized nuclear structure. But the 50% whatever the sperms are formed, they did not have this specialized structure. So Henking was he got a curiosity what it is actually anta. Henking gave a name to this structure as a X body. A specialized nuclear structure ge avaru X body anta hesarisತಾರೆ. X body of Henking was later named as X chromosome. Okay, so the X body was later named as X chromosome. So he got to know that some of the sperms got X chromosome, some of the sperms got Y chromosome. He observed only X chromosome. Why? Because X chromosome is long compared to the Y chromosome. So Y chromosome alliro sperms Y chromosome ಯಾಕೆ ಕಾಣಲಿಲ್ಲ ಅಂತಂದ್ರೆ Y chromosome might be small. So ಆ ರೀಸನ್ಗೂ ಅವರು Y chromosome ಮೇಲೆ ಕಾನ್ಸಂಟ್ರೇಟ್ ಮಾಡಿರ್ಲಿಕ್ಕಿಲ್ಲ. Next, so we have sex determination of grasshopper. These are very important for board actually. ಈ grasshopper ಆಗಿರಬಹುದು, insects ಮತ್ತೆ mammals ಆಗಿರಬಹುದು. For 3 marks they may ask this question. So sex determination of grasshopper ಗೆ ಬಂದಾಗ, grasshopper ಅಲ್ಲಿ how we will recognize that it is a male, it is a female ಅಂತ ಬಂದಾಗ, in a grasshopper it is XXXO type or X0 type. What is this XXX0 type? All egg bears one X chromosome along with autosomes. All egg means female. Okay, they will be having one X chromosome along with autosomes. Some sperms 50% bear one X chromosome, 50% will not bear any chromosomes. So egg fertilized with X chromosome become female. Egg fertilized with sperm without having any chromosome, that is without having X chromosome, X0 type, XO type ಅಂತ ಅಂದಾಗ, that will be regarded as, or that will develop into male. So in the female, the autosomal, the chromosomal complement ಏನಿರುತ್ತೆ, they will ask chromosomal complement. What do you mean by chromosomal complement? Chromosomal ಏನಂತಾರೆ, what we can say, ಹೇಗಿರ್ತವೆ ಕ್ರೋಮೋಸೋಮಲ್ ನಂಬರ್ ಅರೇಂಜ್ಮೆಂಟ್ ಹೇಗಿರ್ತವೆ. ಸೊ 22 ಆಟೋಸೋಮ್ಸ್ ಆದ್ರೆ 23rd ಪೇರ್ ಏನಿರುತ್ತೆ XX ಇದ್ದಾಗ ಇಟ್ ವಿಲ್ ಬಿ ಫೀಮೇಲ್, 22 ಪ್ಲಸ್ XO ಇದ್ದಾಗ ಇಟ್ ವಿಲ್ ಬಿ ಮೇಲ್. ದಿಸ್ ಈಸ್ ಇನ್ ಗ್ರಾಸ್ ಹೋಪರ್. ಓಕೆ, ನೆಕ್ಸ್ಟ್ sex determination in insects and males. Sex determination in insects and males. So both male and female has same number of chromosomes. So illi insects matte mammals ge enirtave, they will be having same number of chromosomes. Female have autosomes along with 2X chromosomes. Males will be having autosomes along with X chromosome and Y chromosome. This is nothing but ours. Mammals andre we will come under this only, no? This is called as male heterogamety and female homogamety. See here, male heterogamety. What do you mean by male heterogamety? Just a minute. Male heterogamety, X and Y, 2 different chromosomes are there. Hetero, different. So the gametes will be getting 2 different chromosomes. Some of the gametes or sperms will be getting X chromosome, some of the sperms will be getting Y chromosome. So hetero anta adakke anta heltare, yaralli, male alli. Next, when we come to the female, we females will be having XX chromosome. So both the chromosomes are XX only. So homo means what? Same. All the gametes will get X chromosomes only. That is why we are regarded as homogamety anta. Very important terms. ಅರ್ಥ ಆದಂಗ ಅನ್ಸುತ್ತೆ, ಬಟ್ ಡೆಫಿನಿಷನ್ ನಿಮಗೆ ಬರಿ ಅಂತ ಅಂದಾಗ ಬರೆಯೋಕ್ ಬರಲ್ಲ. ದಟ್ ಇಸ್ ವೈ ಪ್ರಾಕ್ಟಿಸ್, ರೈಟ್? ದೆನ್ ಸೆಕ್ಸ್ ಡಿಟರ್ಮಿನೇಷನ್ ಸ್ಕ್ರಿಬಲ್ ಡೌನ್ ಮಾಡ್ಕೊಳ್ಳಿ ಅಂತ ಅವಾಗ್ಲೇ ಹೇಳ್ತೀನಿ. ಈಗ ನಾನು ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡಿದಾಗ ಅದು ನಿಮಗೆ ತುಂಬಾ ಅರ್ಥ ಆಗಿದ್ರೆ, ಕನ್ನಡದಲ್ಲಿ ಅರ್ಥ ಆದ್ರೆ ಯು ಜಸ್ಟ್ ಸ್ಕ್ರಿಬಲ್ ಡೌನ್ ದ ಡೆಫಿನಿಷನ್ ಅಂಡ್ ಏನ್ ನಾನು ಕನ್ನಡದಲ್ಲಿ ಏನ್ ಟ್ರಾನ್ಸ್ಲೇಟ್ ಮಾಡಿರ್ತೀನಿ ಅದನ್ನು ನಿಮ್ಗೆ ಅರ್ಥ ಆಗೋ ತರ ಬರ್ಕೊಂಡ್ರೆ ಕಾನ್ಸೆಪ್ಟ್ ಅಂಡರ್ಸ್ಟ್ಯಾಂಡ್ ಆಗುತ್ತೆ. ದಟ್ ಈಸ್ ಮೋಸ್ಟ್ ಇಂಪಾರ್ಟೆಂಟ್. ಓಕೆ, next, when we come to the sex determination in birds, birds ಗೆ ಬಂದಾಗ female birds will be having 2 different sex chromosomes designated as Z and W. In birds, 2 different sex chromosomes are there. One is Z and one is W in female. Male birds will be having ZZ. So here you can observe female heterogamety, male homogamety. I hope you understood, my dear girls. Female heterogamety ಅಂತ ಅಂದ್ರೆ ಯಾಕೆ ಬಂತು? They having different chromosomes Z and W. Male homogamety means what? They have a same chromosome Z and Z. Did you get this? All of you? Yeah? Okay. Fine. I'm sorry. Okay, fine. Just a minute. Next, sex determination in honeybee. So what happens in honeybee actually? Sex determination is honeybee based on number of sets of chromosome an individual receives. How many sets of chromosome that individual receives, no? Based on that, the sex determination will be there. An offspring formed from the fertilization of a sperm and an egg developed into either queen or worker. So an offspring or a honeybee will be formed, which is from the fertilization of egg and the sperm developed into either ಒಂದು ಕ್ವೀನ್ ಆಗ್ಬಹುದು ಅಥವಾ ವರ್ಕರ್ ಆಗಬಹುದು. ಕೆಟಗರಿ ಇದೆ ಹನಿಬೀಸ್ ಅಲ್ಲಿ. And unfertilized egg ಏನಿರುತ್ತೆ, the eggs which are not fertilized by a sperm, so they will be developing into drone or a male by means of parthenogenesis. So parthenogenesis you can observe in honeybee because there the egg directly develops into an individual. The male have half the number of chromosomes than that of female. Actual agi males alli, if female has around, see here, the female are diploid having 32 chromosomes anta iddaga male alli 16 chromosomes irtave. This is called haploid diploid sex determination system. So in which organism you can see this? In honeybee, that is haplodiploid sex determination. If it is diploid, then it is a female. If it is haploid, then it is a male. Male produce sperms by mitosis. They do not have father and then thus cannot have sons but have grandsons. So male produced by sperms by mitosis. Sperms hege produce ಮಾಡ್ತವೆ males mitosis ಮೂಲಕ produce ಮಾಡ್ತವೆ. They do not have father. Why they do not have father? Because they are not fertilized by a sperm. They are unfertilized eggs and thus cannot have sons. So they won't be having the sons at all. Okay. Then, but have grandsons. They will be having the grandsons, they are said. Just a minute. Next, we have all of you understood this statement. Male produce sperms by mitosis. They do not have father and thus cannot have sons but have grandsons. Understood this statement, all of you? Yeah. Please do respond. Okay. Next we have the concept of mutation. Okay, good. So next we have the concept of mutation. What is this mutation actually? It is a phenomenon which results in an alteration of DNA sequences. So just now, while discussing the pleiotropy, the condition which is called phenylketonuria, right? So a phenylketonuria okay. So when we will be we discuss the phenylketonuria disease or a syndrome, in that we got to know the term mutation. So what is that mutation is? It is a phenomenon which results in the alteration of DNA sequences and consequently results in changes in the genotype and phenotype of an organism. So we know that gene is nothing but activated part of DNA. So when there is an alteration in DNA sequences, DNA is made up of nucleotides, alva adenine, guanine. So when there is a change in that alteration of DNA sequences, then it results in the different phenotype, physical expression and different genotype. Okay, that is called as mutation. In addition to recombination, mutation is another phenomena that leads to variation in DNA. So what is this variation is? Recombination antandre crossing over. So due to the crossing over, the variation will occur in the offsprings. Variation andre the differences between the parents as well as children, right? So recombination jotege ee mutation anno concept enide, anno phenomena enide, that will also bring about the what we can say variation. The best example is our mosquito. So ಈಗ mosquito na ಜಾಸ್ತಿ mosquito ಇದೆ ಅಂತ ತಿಳ್ಕೊಳ್ಳಿ. So first they used to put DDT powder for the mosquito. To kill the mosquito ಏನ್ ಮಾಡ್ತಾ ಇದ್ರು, DDT powder ಅನ್ನ ಹಾಕ್ತಾ ಇದ್ರು ಅಥವಾ ಯೂಸ್ ಮಾಡ್ತಾ ಇದ್ರಂತೆ. So at certain, after some years ಅಲ್ವಾ, even though we put DDT powder, the mosquitoes will come. It will not at all affect the mosquito. Why? Because due to the DDT powder, the mutation occurred in the mosquito's genes, so that they will be adapted. They will get adapted. They got adapted to the DDT. Okay, ಆಮೇಲೆ ಏನ್ ಜನರೇಷನ್ ಬರ್ತಾ ಹೋದು ಮಸ್ಕಿಟೋದು, so they got adjusted with the DDT, so that we couldn't kill it by DDT at all. Okay, so the mutation occurs. Mutation will not occur suddenly. ಈ ಒಂದು mutation ಆದ್ರೆ next generation ಆಗಿ ಅಂತ, it takes a lot of time. Okay, next. Loss or deletion or gain or insertion of a segment of DNA results in alteration in chromosomes. Just a minute. Yeah. Loss or deletion. So, ಯಾವುದಾದ್ರೂ ಒಂದು ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ DNA sequence ಅಲ್ಲಿ ಡಿಲೀಟ್ ಆದ್ರೆ ಅಥವಾ ಒಂದು ಎಕ್ಸ್ಟ್ರಾ ಗೇನ್ ಆದ್ರೆ of a segment of DNA results in alteration in chromosomes. Chromosomes ಅಲ್ಲಿ alteration ಆಗತ್ತೆ. Since genes are located on the chromosome, alteration in chromosome results in abnormality and aberration. So we know that genes will be present on the chromosomes. So when there is a problem, or when there is a, what we can say, alteration in the chromosomes, we will be having abnormalities in the genes also. Chromosomal aberrations are commonly observed in cancerous cell. Mutations arise also due to single base pair of DNA. This is called point mutation. So point mutation anta ide. One single nucleotide kuda change adre, it is called as point mutation. Idakke best example anta andre sickle cell anemia, which we will discuss later. Deletion and insertion of base pairs of DNA causes frame shift mutation. If only one nucleotide got changed, then it is called point mutation. So deletion and insertion of base pair idi base pair change adre, then it is called as frame shift mutation anta karitare. Point and frame shift mutation. Understood this, all of you? Yeah, artha aita? Explanation is, yeah, did you get? Okay, fine. Next we have genetic disorders. Just a minute. Okay. Genetic disorders. Pedigree analysis anta baratte. Pedigree analysis. Analysis of traits in several of generations of a family is called as pedigree analysis. Very interesting, actually. Confuse agate, but it is a very interesting study. So analysis of character. Iga one family na consider madana. For example, nimde family athwa nam family anta consider madi. So here, analysis of characters in several of generations. Namma generation. For example, let us take our grandfather, father, and me. So analysis of character from your grandfather to your child. So that is called as pedigree analysis. What we will do in pedigree analysis? In pedigree analysis, the inheritance of a particular trait is represented in the form of, in the family tree over generation. So the inheritance, how a transfer of one character occurred from generations together. For example, one family will be there, those family people will not spend money at all. Okay, ಜಿಪುಟ್ರು ಅಂತ ಹೇಳ್ತಾರೆ, ಜಿಪುಂಡ್ರು ಅಂತ ಹೇಳ್ತಾರೆ. ಸೊ ಆ ಫ್ಯಾಮಿಲಿಯಲ್ಲಿ ಅದೇ ಒಂದು ಗುಣ ಕ್ಯಾರಿ ಆನ್ ಆಗ್ತಾ ಬಂದ್ರೆ, ಸೊ ದಟ್ ಪರ್ಟಿಕ್ಯುಲರ್ ಕ್ಯಾರೆಕ್ಟರ್ ಒಂದು ಫ್ಯಾಮಿಲಿ ಟ್ರೀಯಲ್ಲಿ ರಿಪ್ರೆಸೆಂಟ್ ಮಾಡಿದ್ರೆ ಅದಕ್ಕೆ ಪೆಡಿಗ್ರಿ ಅನಾಲಿಸಿಸ್ ಅಂತ ಕರೀತಾರೆ. ಇಟ್ ಇಸ್ ಜಸ್ಟ್ ಅನ್ ಎಕ್ಸಾಂಪಲ್, ಅದು ಒಂದು ಕ್ಯಾರೆಕ್ಟರ್ ಆಗಿರೋದಿಲ್ಲ, ಇಟ್ ಡಿಪೆಂಡ್ಸ್. ಸೊ ಹಿಯರ್ ಇನ್ ಪೆಡಿಗ್ರಿ ಅನಾಲಿಸಿಸ್ ಅಲ್ಲಿ ಈ ರೀತಿ ನಾವು ರಿಪ್ರೆಸೆಂಟ್ ಮಾಡ್ತೇವೆ. ಇಫ್ ಇಟ್ ಈಸ್ ಓನ್ಲಿ ಅ square, a normal square, then it is a male. If it is a circle, then it is a female. If it is shaded part, then it is a affected individual. Means you cannot recognize or put it as a male or else female. Next, If we establish a line between this square and circle, it indicates mating, okay, or a sexual intercourse. Then whatever here, see, male, female, again here like this, second one. Offspring in birth order, first and second are generations. Offspring number 2, 1, 2, 2. So see here, this is a generation 1, this is a generation 2. Then if we represent like this, then it is an identical twin. If we represent like this, then it is a non-identical twin. Right? So this is regarding pedigree analysis. Artha aita? So based on this, they will ask you the question in NEET as well as CET. Main ಆಗಿರತ್ತೆ, ತುಂಬಾ important. Practice ಮಾಡ್ತಾ ಹೋಗ್ಬೇಕು. Then, so autosomal dominant ಅಂತ ಒಂದು ಕಾನ್ಸೆಪ್ಟ್ ಬರುತ್ತೆ. Affected individual have at least one affected parent. So if any individual is affected, so ಆ ಒಂದು affected individual ಆಗಬೇಕು ಅಥವಾ individual affected ಇದ್ರೆ, so one of the parent will be affected ಅಂತೆ. The phenotype generally appears every generation. Two unaffected parents only have unaffected offspring. If the male and female are unaffected, then the offspring will be unaffected. ಅಂದ್ರೆ ಯಾವುದೇ ರೀತಿಯ ಒಂದು ಎಫೆಕ್ಟ್ ಅಥವಾ ಪ್ರಾಬ್ಲಮ್ ಇರೋದಿಲ್ಲ ಅಂತ. Traits are controlled by dominant gene. Characters are controlled by dominant genes. Both males and females are equally affected. Traits do not skip generation. So generation, ಈ one generation ಗೆ ಇದೆ, ಇನ್ನೊಂದು generation ಗೆ ಇಲ್ಲ ಅಂತ ಅಲ್ಲ. So they will be, you will observe it in every generation. For example, polydactyly, tongue rolling ability, polydactyly, dactyly means ಒಬ್ಬೊಬ್ಬರಿಗೆ 6 ಬೆರಳುಗಳು ಇರ್ತವೆ. ಟಂಗ್ ರೋಲಿಂಗ್ ಎಬಿಲಿಟಿ, ಈ ರೀತಿ ಹಂಗೆ ರೋಲ್ ಮಾಡ್ತಾರೆ. ಐ ಕಾಂಟ್ ಡೂ ಇಟ್. ಸೊ ಅವೆಲ್ಲ ಏನು ಜನರೇಷನ್ಗೆ ಟ್ರಾನ್ಸ್ಫರ್ ಆಗ್ತಾ ಹೋಗುತ್ತೆ. ಸೊ ಇದಕ್ಕೆ ಆಟೋಸೋಮಲ್ ಡಾಮಿನೆಂಟ್ ಅಂತ ಕರೀತಾರೆ. See, this is the affected individuals. Both the affected individuals are there. So when you see here, 2 children are normal and 1 more children is affected. So when one parent is affected, see their children. 2 children are normal and 1 is affected. Alva? So like that occurs. If the parents are normal, the offsprings will be normal. If only one parent is affected, then any of the child or any of the offspring will be affected. Next we have Mendelian disorders anta, very interesting concept idu. So it is also named as genetic disorders. Genetic disorders alli 2 ವಿಧ ಇದೆ. ಒಂದು Mendelian disorders, ಇನ್ನೊಂದು chromosomal disorders. Mendelian disorders are mainly determined by a alteration or mutation in the single gene. When alteration, only one gene is altered or mutated, any change occurred in that what we can say gene, then it is called as Mendelian disorder. It obeys the principle of Mendelian inheritance during transmission from one generation to other. Means ಈ ಒಂದು Mendelian disorders ಆ Mendelian concepts ಅನ್ನ ಒಬೆ ಮಾಡ್ತವೆ. It can be expressed in the pedigree analysis. Examples ಏನೇನು ಅಂದ್ರೆ hemophilia, color blindness, cystic fibrosis, sickle cell anemia, phenylketonuria, thalassemia. So these all are the Mendelian disorders. Right. So Priyanka, can you read hemophilia? Can you read hemophilia?
It is the disease of a single protein that is a part of the subset of protein involved in the clotting of blood is affected. Due to this affected individual, a simple cut will result in non-stopping bleeding. It is a recessive disease. The disease transmitted from unaffected carrier female becomes a female progeny. Female becoming hemophilia is extremely rare because mother of such a female athlete.
Continue. So because a mother of a female carrier and the father should be hemophilic. Affected transmits the disease only to the son, not to the daughter. Daughter can receive the disease from mother and father. Okay. So here, hemophilia ಅನ್ನೋ ಡಿಸೀಸ್ ಇದೆ. ಈ ಡಿಸೀಸ್ ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ, a single protein which is a part of cascade of proteins, means group of protein which are involved in the clotting of a blood, that is a stopping of the blood, will be affected. So due to this, an affected individual, so ಆ ಒಂದು protein part single, that is a part of affected. Okay, ಆ protein ಒಂದು ಎಫೆಕ್ಟ್ ಆಗಿರೋ ಕಾರಣ, so the individual, if he got a single cut, so there will be a non-stop bleeding. So this is sex-linked recessive disease ante. It is sex-linked recessive disease. The disease will be transmitted from unaffected carrier female to some of the male progeny. If the female is unaffected, but she is a carrier, she has that gene, but she don't have hemophilia, then she acts as a carrier female. ಅಂದ್ರೆ ಆ ಜೀನ್ ಪ್ರೆಸೆಂಟ್ ಇರತ್ತೆ, ಬಟ್ ಆ ಜೀನ್ ಎಕ್ಸ್ಪ್ರೆಸ್ ಆಗಿರೋದಿಲ್ಲ ಫಿಮೇಲ್ ಅಲ್ಲಿ. ಸೊ ಅವಾಗ ನಾವು ಕ್ಯಾರಿಯರ್ ಫಿಮೇಲ್ ಅಂತ ಕರೀತೇವೆ. ಸೊ ಇಟ್ ಮೈಟ್ ಟ್ರಾನ್ಸ್ಮಿಟ್ ಫ್ರಮ್ ಕ್ಯಾರಿಯರ್ ಫಿಮೇಲ್ ಟು ಒನ್ ಆಫ್ ದ ಮೇಲ್, ಅಂದ್ರೆ ಟು ಒನ್ ಆಫ್ ದ ಸನ್ಸ್. ಫಿಮೇಲ್ ಬಿಕಮಿಂಗ್ ಹೀಮೋಫಿಲಿಕ್ ಇಸ್ ಎಕ್ಸ್ಟ್ರೀಮ್ಲಿ ರೇರ್. ಫಿಮೇಲ್ ಅಲ್ಲಿ ಈ ಹೀಮೋಫಿಲಿಯಾ ಅನ್ನೋ ಕಂಡೀಷನ್ ರೇರ್ ಆಗಿ ಕಾಣಿಸ್ಕೊಳ್ಳುತ್ತೆ. ಬಿಕಾಸ್ ಮದರ್ ಆಫ್ ಸಚ್ ಅ ಫಿಮೇಲ್, ಬಿಕಾಸ್ ದ ಫಿಮೇಲ್ ಹೂ ಇಸ್ ಎಫೆಕ್ಟೆಡ್ ಅಲ್ವಾ, mother of such a female, at least carrier and the father should be hemophilic. ಯಾವ females ಅಲ್ಲಿ ಇದು ಕಂಡು ಬರುತ್ತೆ ಅಂತ ಅಂದ್ರೆ, it is usually not observed in the females. If it is observed, that females parents will be there, no? So that parents ಅಲ್ಲಿ mother will be a carrier, father will be a affected, that is he will be having hemophilia. So in that condition, the female may get hemophilia. Affected transmits the disease only to the son, not to the daughter. ಈ affected individuals ಏನಿರ್ತಾರೆ, out of two parents, if one is affected, so that character or that gene will be transmitted, that disease will be transmitted only to the sons ante, not to the daughters. Daughter can receive the disease from both mother and father. As I said, when the mother is a carrier, father is hemophilic, both are affected ಇದ್ದಾಗ, ಅಂದ್ರೆ ಮದರ್ ಅಲ್ಲಿ ಕ್ಯಾರಿಯರ್ ಇದು ಎಫೆಕ್ಟ್ ಆಗಿರಲ್ಲ, ಬಟ್ ಶಿ ವಿಲ್ ಬಿ ಹ್ಯಾವಿಂಗ್ ದಟ್ ಜೀನ್. ಫಾದರ್ ಅಲ್ಲಿ ಹಿ ವಿಲ್ ಬಿ ಎಫೆಕ್ಟೆಡ್, ಅಂದ್ರೆ ಹಿ ವಿಲ್ ಬಿ ಹ್ಯಾವಿಂಗ್ ಹೀಮೋಫೀಲಿಯಾ. ಸೊ ಈ ಕಂಡೀಷನ್ ಇದ್ದಾಗ ಮಾತ್ರ ದಟ್ ಡಿಸೀಸ್ ವಿಲ್ ಬಿ ಅಬ್ಸರ್ವ್ಡ್ ಇನ್ ದ ಡಾಟರ್ಸ್ ಅಂತೆ. ನೆಕ್ಸ್ಟ್ ವಿ ಹ್ಯಾವ್ ಸಿಕಲ್ ಸೆಲ್ ಅನೇಮಿಯಾ. ಸೊ ಇಲ್ಲಿ the defect is caused due to substitution of glutamic acid by valine. So here what happens, the glutamic acid will be substituted by valine at the 6th position of beta globin chain of hemoglobin molecule. So hemoglobin molecule will be having a chain. So ಆ chain ಅಲ್ಲಿ beta globin chain ಅಂತ ಇರುತ್ತೆ. ಆ beta globin chain ಅಲ್ಲಿ 6th position ಅಲ್ಲಿ the substitution of glutamic acid valine by valine ಆಗತ್ತೆ 6th position ಅಲ್ಲಿ. So substitution of amino acid takes place due to single base substitution at the 6th codon of the beta globulin gene from GAG to GUG. If the codon is GAG, then it is glutamic acid. If it becomes GUG, then it is valine. So ಒಂದೇ ಒಂದು codon ಇಲ್ಲಿ change ಆಗಿರೋ ಕಾರಣ ಈ ಒಂದು ಕಾಸ್ ಆಗತ್ತೆ. The mutant hemoglobin, so ಅವಾಗ ಆ ಸ್ಟ್ರಕ್ಚರ್ ಏನಾಗತ್ತೆ, it will become mutant because there is a change in one codon. Hemoglobin molecule undergo polymerization chain reaction ಹೋಗುತ್ತೆ under low oxygen tension, causing the change in the shape of RBC. So usually shape of RBC ಏನಿರುತ್ತೆ, biconcave shape ಇರುತ್ತೆ. So ಅಲ್ಲಿ biconcave ಗಿಂತ to elongated sickle like structure, elongated ಉದ್ದವಾಗಿ ಆಗತ್ತೆ. This is an autosomal linked recessive trait transmitted from parents to offspring when both the parents are carrier. ಎರಡು ಮದರ್ ಮತ್ತೆ ಫಾದರ್ ಇಬ್ರು ಕ್ಯಾರಿಯರ್ಸ್ ಇದ್ರೆ, ಅಂದ್ರೆ they will be having that genes but they don't have that condition, ಅವಾಗ ಅದು ಮಕ್ಕಳಿಗೆ ಟ್ರಾನ್ಸ್ಫರ್ ಆಗೋ ಚಾನ್ಸಸ್ ಇರ್ತವೆ. This disease is controlled by a single pair of allele. So here HBA and HBS. There are three possible genotypes: HBA, 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 HBS, HBS, HBS. Only homozygous individual for HBS show the diseased phenotype. So ಇಲ್ಲಿ ಎರಡು ಹೋಮೋಸೈಗಸ್ ಇಂಡಿವಿಜುವಲ್ ಇದ್ರೆ, ಎಸ್ ಅಂತ ಎರಡು ಹೋಮೋಸೈಗಸ್ ಇಂಡಿವಿಜುವಲ್ಸ್ ಇದ್ರೆ, ದೆನ್ ಆ ಡಿಸೀಸು ಫಿನೋಟೈಪಿಕ್ ಎಕ್ಸ್ಪ್ರೆಷನ್ ತೋರ್ಸತ್ತಂತೆ. ಹೆಟೆರೋಸೈಗಸ್ ಕಂಡೀಷನ್ ಅಲ್ಲಿದ್ರೆ HBA, HBS ಅಪಿಯರ್ ಅಪರೆಂಟ್ಲಿ ಅನ್ಎಫೆಕ್ಟೆಡ್, ಅಂದ್ರೆ ಅವರಿಗೆ ಸಿಕ್ಕಲ್ ಸೆಲ್ ಅನೇಮಿಯಾ ಇಲ್ಲ ಅಂತ ಅನ್ಸತ್ತೆ, ಬಟ್ ದೇ ವಿಲ್ ಬಿ ದ ಕ್ಯಾರಿಯರ್ ಆಫ್ ದ ಡಿಸೀಸ್ ಅಂತೆ. ಸೊ ಹಿಯರ್ ಯು ಕ್ಯಾನ್ ಸಿ, ಇಲ್ಲಿ ನೋಡಿ, DNA ನಾರ್ಮಲ್, ಇದು ಸಿಕ್ಕಲ್ ಸೆಲ್. ಓಕೆ, DNA GAG, G will codes for C, A will code for T, G will code for C. So like that RNA ನೂ ಇದೆ. ಸೊ RNA ದಲ್ಲಿ GAG ಆಗತ್ತೆ. ಸೊ ಇಟ್ ವಿಲ್ ಕೋಡ್ ಫಾರ್ ಗ್ಲುಟಮಿಕ್ ಆಸಿಡ್. ದಿಸ್ ಇಸ್ ಅ ನಾರ್ಮಲ್ ಪ್ರೋಟೀನ್. ಸಿಕ್ಕಲ್ ಸೆಲ್ ಅಲ್ಲಿ ಏನಾಗತ್ತೆ, GAG ಬದಲು GUG, GTG ಬರುತ್ತೆ. GTG ಇಲ್ಲಿ ಕೋಡ್ ಆದ್ರೆ ಹಿಯರ್ ಇಟ್ ವಿಲ್ ಕಮ್ CAC. CAC ಇಲ್ಲಿ ಬಂದ್ರೆ it will come GUG. GUG will code for valine. So it is a mutant protein or mutant, we can say, amino acid. So ಈ ರೀತಿ valine ಬಂದಾಗ there will be change in a structure of a hemoglobin which will lead to the change in shape of RBC. ಅದಕ್ಕೆ ಸಿಕ್ಕಲ್ ಸೆಲ್ ಅನೇಮಿಯಾ ಅಂತ ಕರಿತೇವೆ. Yeah. Komala, can you read this phenylketonuria? Komala is not here. Okay. Aishu, can you read this? ನಿನ್ನ ಮೈಕ್ ಮ್ಯೂಟ್ ಅಲ್ಲಿ ಇದೆ ಅಮ್ಮ, ಮೈಕ್ ಮ್ಯೂಟ್ ಅಲ್ಲಿ ಇದೆ.
The autosomal recessive trait inborn error of metabolism. The affected individual lack one enzyme called phenylalanine hydroxylase that convert the amino acid phenylalanine to tyrosine. Okay. In the absence of the enzyme, phenylalanine accumulated and converted into pyruvic acid and other
derivatives.
Accumulation of these results in mental
retardation.
This derives excreted through kidney.
Okay, thank you. So phenylketonuria dalli ಏನೈತೆ, this is also autosomal recessive trait. It is an inborn error metabolism. It will come by birth. The affected individual lack one enzyme. ಯಾವ ಒಂದು ಇಂಡಿವಿಜುವಲ್ ಗೆ ಇದು ಎಫೆಕ್ಟ್ ಆಗಿರುತ್ತೋ ಅವರಲ್ಲಿ ಒಂದು ಎಂಜೈಮ್ ನ ಕೊರತೆ ಆಗತ್ತೆ. ವಿಚ್ ಇಸ್ ದಟ್ ಎಂಜೈಮ್? phenylalanine hydroxylase. ಲ್ಯಾಕ್ ಮೀನ್ಸ್ ಕೊರತೆ ಅಂತ. phenylalanine hydroxylase ಅನ್ನೋ ಎಂಜೈಮು ಅವರಲ್ಲಿ ಕೊರತೆ ಇರುತ್ತೆ. ಸೊ ಅದಿದ್ರೆ ಏನಾಗ್ತಿರತ್ತೆ, ದಟ್ ಕನ್ವರ್ಟ್ಸ್ ಅಮೈನೋ ಆಸಿಡ್ phenylalanine ಟು ಟೈರೋಸಿನ್. ಸೊ ವಾಟ್ ಈಸ್ ದ ಫಂಕ್ಷನ್ ಆಫ್ ಇಟ್? ಇಟ್ ಕನ್ವರ್ಟ್ಸ್, ಅದು phenylalanine ಇರೋದನ್ನ ಟೈರೋಸಿನ್ ಗೆ ಪರಿವರ್ತನೆಯನ್ನ ಮಾಡುತ್ತೆ. ಇಫ್ ದಿಸ್ ಎಂಜೈಮ್ ಇಸ್ ನಾಟ್ ದೇರ್, ಸೊ ಅವಾಗ ಏನಾಗುತ್ತೆ, ದೇರ್ ವಿಲ್ ಬಿ ಅಕ್ಯುಮಿಲೇಷನ್ ಆಫ್ phenylalanine ಕನ್ವರ್ಟ್ ಆಗದೆ, ಪರಿವರ್ತನೆ ಹೊಂದದೆ ಹಾಗೆ ಉಳಿತಾ ಹೋಗುತ್ತೆ, ಶೇಖರಣೆ ಆಗುತ್ತೆ, ಅಕ್ಯುಮಿಲೇಷನ್. ಅಂಡ್ ಇಟ್ ವಿಲ್ ಬಿ ಕನ್ವರ್ಟೆಡ್ ಇಂಟು phenylpyruvic acid and other derivatives. Instead of tyrosine, it will be converted into phenylpyruvic acid and other derivatives ante. Accumulation of these results in mental retardation. So yavaga in the blood the phenylpyruvic acid and other derivatives will accumulate alva. So this will result in mental retardation. Okay, so this is regarding phenylketonuria. Next, we are left out with chromosomal disorders. Here we have Down syndrome, Klinefelter syndrome and Turner syndrome. Three concepts are left out, which I will be explaining in the upcoming session at 3 o'clock. Okay, please stay tuned in. Don't go anywhere. After completion of this, we will be discussing multiple choice questions of NEET. Okay. fine. Before that, at 3 o'clock when you attend the session, please be ready with your textbook. Textbooks na ನಿಮ್ಮ ಹತ್ರನೇ ಇಟ್ಕೊಂಡು ಕೂತ್ಕೊಂಡಿರಿ ಆಯ್ತಾ. Okay, let me end up the session here. Have a nice day. Take care. Bye bye.